Alright students, time for a quick look at the last section in Chapter 4, which is Advances in Genetics, and starts on page 123. So, look real quick at selective breeding. This, I would say, was an advance in genetics probably 10,000 years ago when humans first started cultivating animals, raising plants that we chose, and when you choose, you can choose the best ones. So selective breeding is a great way to give us the very best that we want. And that's how in 1930, when we started breeding turkeys or keeping track of their weight, your average turkey weighed around 13 pounds. And today, 80 years later, your average turkey weighs just under 30 pounds. So that's more than doubled the average turkey weight just by only picking the best organisms, the biggest, fattest turkeys, and breeding them together. So when we've got a trait that we're looking at like that, where we want both parents to have the trait, it's called inbreeding. Now this is not related to whether or not those turkeys are family members or not. Inbreeding strictly refers to these animals having the same desirable trait. They may be family members or they may not. It's not important to us. Breeders want to avoid actual inbreeding of family members as much as possible so you don't end up with too many negative alleles building up. So, in breeding, we breed animals with similar traits, hoping that the offspring will also have those traits, maybe even better. Hybridization is kind of the opposite of that. I'll have one animal that has a positive trait, another animal that has a different good trait, and I'm going to breed them together, hoping that the babies will have both positive traits. So I may have a fruit tree, one fruit tree produces good, tasty fruit, but it gets diseases very often. The other fruit tree may produce not too tasty fruit, but it resists real cold winters and it resists all the diseases. So in hybridization, I'm going to cross two different animals, or plants, or funguses, if that's your thing, and hope that the offspring gets the best of both parents. So hybridization is crossing two different animals, trying to get both good traits into the offspring. Of course, it's always chance, because this is just plain old breeding. So you could get a nasty tasting fruit tree with lots of diseases and stuff like that. So it can go wrong, but there's also odds of you getting the ones you're looking for. So those are the standard breeding techniques. Next is cloning. Cloning, there's a lot we can say about it, but really the idea is very, very simple. You take the egg of an animal and empty it out, and we take the nucleus of a donor animal, that's all the code that tells how to make him, put it in that egg, and then we implant the egg into a surrogate mother. The surrogate mother will grow the baby out, and it'll be born, but it'll be born as the creature that the donor was. So whoever we got that nucleus from, that's going to be who the baby is. So remember, this isn't actually breeding. There's no fertilization and no meiosis going on. We're producing an exact genetic copy of the first animal. So, if using selective breeding, we ended up with a really good animal, and we didn't think it was likely we could do that again, we could clone that animal and get many, many more just like him. And that's a big advantage because we don't have to deal with the uncertainties of plain old breeding. We know we're going to get the animal we want every single time. So that is cloning. Next is genetic engineering. This is a very, very broad topic because there is so much that we can do that will be a big help to all humanity if we're careful and we do it right. So genetic engineering means that we take a gene, or maybe many genes, from one animal or plant or other organism and put them into another organism. So we can swap genes from bacteria into animals, put animal genes into plants, and all kind of mixed up stuff. So genetic engineering gives us a lot of advantage. First example is genetic engineering in bacteria. Recall that we took the human gene for insulin, and everybody knows that people with diabetes need insulin. It's very important to help them survive. So we can put that gene into bacteria, so it's still the human copy of the gene, it still makes perfect human insulin, but now it's going to be produced by a bacteria that we can grow easily in vats and produce large amounts of this very valuable medicine. So genetic engineering lets us add genes to other organisms that may produce the medicines we need much more efficiently. So that's the example for bacteria, is when we place the insulin gene into bacteria to produce human insulin. Next example would be genetic engineering in other organisms, which is quite a bit more tricky to perform than genetic engineering in bacteria, but scientists have still gotten quite good at it. So in genetic engineering in other animals, we can produce cows or goats that produce other animal proteins in their milk. We have made a goat that produced spider silk protein. Now, it didn't work out as expected. We didn't get a product that we could use to make spider fibers, but the protein was definitely in the animal's milk. Just didn't work out exactly like we'd hoped. But we could also make human genes go into a cow or a goat and have that animal produce medicine, which would be a great advantage for people far, far from medical care where you can't get to a pharmacy. You could milk your animal for your medicine, which would be a big advantage from having to buy it and have it shipped from another country far, far away. So it'd be a great advantage for poor people or people who live in remote villages. We could also change plants so that they'll grow in poor or salty soil, or grow in places where it's very, very dry, or grow in places where it's very, very cold. And this would also help people in the more troubled parts of the world because they could grow their own food even though their land is very, very poor quality, and be able to provide for themselves and not have to depend on the rest of the world for their livelihood, which would be a great advantage. Freeing people from those hardships would be a great thing to do with this kind of technology. So that's genetic engineering in other animals. Of course, there's really no limit. I showed you plenty of pictures of fluorescent animals and things like that. So we see there's a lot we can do and a lot of uses for this technology. So there are some concerns about it, and lots of times we don't expect a long-term effect, and then we get one. And I talked about Roundup resistance. Now, we can give corn plants and other plants a gene called Starlink that will prevent Roundup from killing that plant, which is great for farmers because Roundup is a very safe herbicide. It reduces your need for fertilizer. It reduces the amount of work the farmer has to do, so it's a really great plan. But when we were spraying that much Roundup, eventually a wild plant got a mutation that let it defeat the Roundup also. So soon we had lots of this plant called horserush, which was resistant to Roundup, growing in Roundup-treated fields. And we could probably expect to see more of that happen as we continue to use Roundup like that. So that was an unexpected effect. And of course, you got to think about other unexpected effects, like people being allergic to certain chemicals made by the genes we add to these. So we need to consider all these things closely. Scientists now talk to farmers to make sure that we understand how it's going to affect farm culture, talk to other people, talk to environmentalists, and make sure we can understand what all the consequences of adding these genes may be, because we may have missed some. Now, genetic engineering could be very powerful. Now, learning more about human DNA is going to take us even further into the future. The Human Genome Project has just been completed, and now they're trying to add many, many more humans to this project. We got just a handful of people sequenced now, but if we can get that number into multiple thousands of human genomes, we'll really start to learn about what it is going on in humans. So the Human Genome Project here has now shown us the DNA sequence of every single human gene in those couple of people. This is a big advantage because if you have a damaged copy of a gene, we can look it up in the gene database, find what the good gene looks like, and later in the future we'll probably be able to use gene therapy to use that copy from the gene database to heal you. That's the idea behind the Human Genome Project, apart from helping us learn how humans are put together and how we work and when disorders occur, what's going on. It'll help us fix people who are having troubles. So the Human Genome Project, let's remember, very important, will be on the test, has shown us every sequence of every gene in the human genome. That's something like 6 billion letters, A, C's, T's, and G's, so that is a lot of data. DNA fingerprinting is a useful technique, and you've probably seen this on TV quite a bit, but maybe not as accurately as it occurs, is a technique that lets us see how unique every single human is. This is not related to the fingerprints on your fingers. We only call it that because they're unique, and this process is also unique. So it lets us see exactly what a person is and how different they are from another person. What happens is we take your DNA and apply genetic scissors and cut it up in several unique ways. Every person's DNA will cut up in a different way, so they'll all have different lengths. We sort those pieces of DNA by their length, shorter ones from the longer ones, and we'll get unique patterns that should not match anybody else unless they are identical twins. So if we do enough different cuts, it gets us about a 1 in a billion person chance of getting the wrong result if we're doing it right. So DNA fingerprinting can help us identify criminals from trace evidence left at a crime scene, can help us identify missing persons, or people who are, say, lost in war, be able to identify their remains, and also to identify family relationships, identify long-lost family members, and in the case of a baby mix-up at the hospital, we can identify which parent should have which baby. Of course, that doesn't really happen that often, but if it did happen, this would be a technique we could use. So that's a quick look at advances in genetics. Remember, some of these are more advanced than others, and this is an area that is really moving fast. Oh, I didn't mention gene therapy. We had talked about it, but under genetic engineering, gene therapy is a very good way of adding a gene to a person if their gene is damaged. So if I have a damaged cystic fibrosis gene, so I have the condition cystic fibrosis, genetic engineering can put that gene into a virus. I can inhale that virus, but instead of making me sick, that virus will give me the good copy of the gene and I will be temporarily healed. Genetic engineering is working very well these days, and we expect to see a lot more techniques using it in the future. So, that is Advances in Genetics, and that's the last section in Chapter 4.